Good morning, everybody. Welcome to this week's Ausenco Connect webinar. We will begin in 1 minute. So good morning, folks from around the world. Well, good morning from Vancouver, where it's 9:01 a.m. Welcome to Ausenco Connect. This is Ausenco's bi-weekly webinar series, bringing together Ausenco subject matter experts to our clients, where we can continue to learn and engage with each other during COVID. My name is Scott Weston, and I will be your moderator for this morning. I'm personally very excited about today's topic and our presenter, so you're in for a treat. Today's topic is transforming environmental surveys for major projects with eDNA. And eDNA, or environmental DNA, is a new and emerging, well, it's not new, it's an emerging tool used for identifying species in the environment. And it's something that we at Ausenco have been able to deploy and bring great value to our clients. And Dr. Doug Bright, who is your who is your presenter this morning, is somebody who's been working many years on this, and is one of Western Canada's foremost leaders in this subject. Doug has a graduate degree based on a research focus on environmental toxicology and marine ecology. He has been working on mining-related environmental issues since 1985. And after 11 years in applied research in a university setting, Doug moved to the private sector position in 2002. He's been with Hemmera since 2011, leading the environmental risk assessment services. He's passionate about continuing learning and innovation and collaboration. So in a moment, I'll hand over to Doug. My name is Scott Weston. I'm the Vice President of Business Development at Hemmera, which is the environmental arm of Ausenco in Canada. And as I said, I'm your moderator. During the session, please write in any questions you have in the Q&A box. It should be on the right side of your screen, and if it's not, go down to the ribbon near the bottom and click on the question mark Q&A circle, and the dialog box will pop up on the right. Write your questions in there during the session, and when Dr. Bright is finished with his presentation, I will come back on and moderate the Q&A session with Doug. So please put your questions in during the session as they arrive. So with that, I'm going to hand over to Dr. Doug Bright for transforming environmental surveys for major projects with eDNA.
Great, thank you, Scott, and good morning. Lori, let's just start with the title slide, please. Yeah, we appreciate your taking time out from your busy day-to-day to talk about innovative ways for improving how we conduct environmental impact assessments, environmental effects monitoring, and cumulative effects assessments. Because I'm old and I actually get center stage, I'm going to basically be a bit abusive of your time and pontificate a little bit. So my worldviews are shaped a little bit by my age. I grew up in the 60s and have been around for a few decades. So let's talk about sustainability. The report entitled Our Common Future was produced by the World Commission on Environment and Development in 1987. That's the so-called Brundtland Commission report. That's not the first time that Western society and humans in general have basically tried to advance the ecologically and socially sustainable economic development in the post-World War II era, but it's kind of the first time it really stuck and has kind of slowly gained traction ever since. The Brundtland report was written some 35 years ago. So we've got 35 years of history since. That particular report, the Brundtland Commission, touched on a bunch of things, including poverty reduction worldwide, but in particular, it's known for really defining the three pillars of sustainability: environment, economic, and social. At present, there are many voices worldwide calling for green recovery. You'll have heard some of this stuff in the news following the COVID pandemic. We see signs during COVID, ironically, of better air quality, less amounts of noise in the marine environment and things like that. We have many voices calling for activities and projects that are net zero coming out of COVID and going forward in the future in general, and lots of voices that are basically calling for the protection and enhancement of biodiversity, however one might define that. Between these, let's say, 60s and 70s or 80s and now, from my perspective, what has changed dramatically is not in the number of voices as much as in how global investors, central banks, pension funds, governments, and the collective population are beginning to take very seriously environmental and social aspects of all of the human activities, new projects, existing activities, monitoring, and of all those kinds of things. So that's a bit of context. Next slide, please. So that's kind of a relatively long-winded way of saying that ecological protections and improvements matter, perhaps now more than ever in the history of humankind. So by extension, a good grasp on what's out there and on the ecosystems and what is in need of protection and how our activities are working in terms of protecting and enhancing things like biodiversity and ecosystem functions becomes important now more than ever. Next slide, please. So this is really kind of gravitating towards ecology. But my hope today was not to actually provide basically a talk by an ecologist for ecologists, but to talk more broadly about environmental needs in the context of managing human activities. We do need to talk a little bit about where we've come from. And so I want to talk a little bit about traditional ecological surveys and their limitations. And so when I think about traditional ecological surveys, I'm thinking about fish surveys where we might go out and do electrofishing or deploy minnow traps or various other techniques, or wildlife surveys where we're basically listing for bird calls and amphibian calls and things like that, or time-constrained searches and those kinds of things. If you kind of look at all of those techniques altogether, a couple of things emerge. One of the ones is that those kinds of ecological surveys, the traditional ecological surveys, really rely on some very specialized biological, ecological knowledge. So there's a professional reliance there. There is an inherent bias in relying on professional expertise. One of the things that we as decision makers have gotten quite used to, and we kind of ignore now, is that traditional ecological surveys tend to have low probabilities of detecting species of management interest if they're indeed there in the environment, particularly if, for example, a wildlife or fisheries biologist goes out to a site for limited time periods, once, for example, as opposed to multiple times, or over a few hours as opposed to many days. It decreases the probability of actually finding something that might be important. One of the reasons that we tend to spend less effort out in the field as biologists than we'd like to is that traditional ecological surveys tend to be time and labor intensive. And another important aspect of traditional ecological surveys is they intend to be intrusive of the habitat that we're trying to protect. In some cases, we're turning over rocks in stream beds or walking up stream beds, and it requires basically intimate kind of interactions with the kind of the ecosystems and habitats that we're surveying. That in turn increases the risk of greater transmission of things like wildlife pathogens, which can have obvious negative consequences. Next slide, please, Laurie. What I'd like to talk about for the rest of this talk is talk about environmental DNA as an innovative approach to overcome some of those problems and create some what I think are some really important benefits moving forward. So what I'd like to talk about is environmental DNA. What is it? What's it really useful for? What are its limitations, and where should we be cautious about interpretations based on eDNA as a line of evidence as opposed to other line of evidence? And then I'd like to provide some concrete examples. Next slide, please. Before I jump into it, one of the things about eDNA is, Hemmera has been doing eDNA studies for the last 5-6 years, but it's not about what Hemmera does. eDNA is something that has increased exponentially within the academic research community since 2008, when basically the first real proof of concept was out there for things other than microbial ecology. And if you haven't heard of the term and are not terribly familiar with the term now, it's something that, whether we like it or not, we're going to see more and more and more of, not just in basically the academic research sphere, but in terms of work being done in Canada right now and around the world by government research scientists, by private sector organizations, by laboratories that are getting set up to actually do the lab portions of eDNA work, and so on and so forth. So basically the train is coming down the track one way or the other. Next slide, please. So environmental DNA. So in its kind of simplest form, environmental DNA is simply DNA, the genetic code of life for an organism or a group of organisms that basically is shed into an environmental sample. And eDNA techniques involve collecting environmental samples and evaluating what is in that environment based on the DNA in that particular sample. So that's it in a nutshell. Obviously, the technique works just like human forensics, because each species or each kind of groups of species have unique aspects to their genetic code, a little bit like barcodes, as when you run your groceries through the till at the shopping center, that allow us to actually, after the fact, take the DNA and identify what species contributed the DNA to that environmental sample. A lot of what we're capable of doing now as scientists is facilitated by the barcoding of big chunks of all of the species that are out there on the planet, be they, whether they're fungi or invertebrates or plants, in particular based on the International Barcode of Life project. Next slide, please. So again, eDNA studies at their heart are actually really simple. So if eDNA is basically DNA basically contained within an environmental sample, then the studies, it follows that the studies are basically, you got to think about what you're doing and what the species ecology is and so on and so forth. Like anything else, you need to have a good survey and study design. We go out and we collect an environmental sample, whether that's a sample of soil or sediment, or in some cases filtered air samples, or most typically water samples. We isolate the DNA or the material containing the DNA from that particular sampling medium. We take that medium into the laboratory, or sometimes we do it in the field. We further isolate the DNA from that sample. We extract the DNA. We analyze that DNA through a process of amplification, most typically in technology called quantitative polymerase chain reaction type of machines. And then we interpret the results towards an understanding of the kind of organisms that may have contributed the DNA. Next slide, please. So this is an ugly study. I appreciate that. So the point being is that, the kind of steps we talked about, from basically environmental sample collection all the way through to interpretation, is common to all eDNA studies. But just for everybody to be aware that eDNA studies actually comprise a wide range of techniques. In some cases, for example, when we're doing microbial ecology, for example, on mine spoils, we're actually analyzing the DNA that comes from living microbial cells. So in some cases, the applications involve really extracting DNA from living organisms. An example application of that, for example, is if we take stream invertebrates using kick netting, as we would in the Canadian Aquatic Bioassessment Monitoring Program, the CABIN programs, and actually took all of the larval insects that we got out of that sample and mushed them up and ran them through qPCR. That's an example of using eDNA that's actually was living within in an organism to reconstruct the community in terms of presence absence. In many cases, we're looking at shed DNA, DNA that's already been lost to the water column, or actually may end up in the sediments. There are techniques, because DNA can be preserved in anoxic sediments for long periods of time, where we can construct the long-term ecological history of a watershed based on the DNA that's preserved over literally hundreds of years as you go down through the sediment profile. There are targeted taxa methods where we can actually look for a single species and incredibly sensitive, and there are a number of methods out there where we're actually looking for everything that was basically that is possibly around at the kind of community level. So those kind of community level approaches are sometimes referred to as barcoding or metagenomics approaches. Next slide, please. And again, that may seem a little bit overwhelming, but just be aware that when people talk about eDNA studies, they may be talking about a number of different things, and there are some specific considerations for all of those kinds of things. So what are the advantages of eDNA? First and foremost, I guess there's a number of advantages, but amongst the advantages is that eDNA techniques are incredibly effective. If I collect a water sample from some location, 1 liter or a 5 liter water sample, and filter it, all I need is really one copy of DNA of an organism to actually get a positive result when I actually run it through the rest of the process. And so the sensitivity of eDNA is well recognized within the scientific community, and it's a really good technique in particular for trying to do censuses and surveys of those species that are very cryptic and very rare, because I can detect them at a stage where other traditional techniques can't detect them. eDNA has the potential to be more accurate than traditional ecological surveys. It involves less bias, as I talked about, but it starts to bring biological, ecological observations into the same kind of realm that we actually have come to recognize for environmental chemistry. So they're called use labs that actually have the appropriate certifications. We can develop operating procedures, and we can actually increase reproducibility of the data for DNA studies far more than for ecological survey approaches, and increase the reliability overall. We can actually synchronously study multiple species instead of one species at a time. You might imagine in a traditional ecological survey approach, we might actually have to send out an amphibian specialist and an avian specialist and a fish specialist. And depending on the circumstances, we can actually send out appropriately trained people and do studies of a wide range of taxa using the same kinds of samples. One of the advantages of eDNA is that once we've actually captured the eDNA sample and extracted it from the environment sample, we can actually archive that sample. So we can go back many years later and say, okay, I'm not really sure if that disease was in the wildlife population, or I'm not really sure if that was an endemic species to the area. In fact, we can take that eDNA, and with the development of new assays in the future, we can take the sample and rerun the sample and say, okay, let's look at additional species in that particular sample. So we can do retroactive evaluations. And because we're collecting just doing simple sampling of environmental media, we tend to be less invasive of the habitats that we're sampling in. Next slide, please. So the biggest advantage of all of eDNA approaches over traditional ecological type approaches really has to do with the level of effort to get to a set of information and a set of knowledge. And there's kind of lots of estimates out there, but fundamentally, a good generalization is that I can spend 1/10 the amount of effort to get the same amount of information using eDNA as opposed to using kind of traditional ecological methods. So, and I'll give some examples of that later on, but what that basically means now is that it's an enabling a mechanism for people to actually gather more information within the ecosystems and landscapes that they're operating. So that in turn gives the potential to fill in the holes in terms of where we're missing data on different kinds of species. It allows us to actually conduct our studies over greater scales and over more extensive parts of the watershed. It allows us to go back more routinely and get really good temporal trends analysis for some types of observational data. So what we end up doing then with eDNA is having an enabling mechanism that leads us to big data approaches for dealing with ecosystem type issues. For me, that's the excitement of eDNA. Next slide, please. We'll skip this one, Lori. Next slide, please. There's some limitations of eDNA. There's some real nuances and subtleties about how you do it and whether or not the data you produce are credible. But fundamentally, this is true of all DNA studies, is that the current stage of development for eDNA means we can basically collect a sample and run it through all of our procedures, and we get presence absence data. So we can tell from our sampling and our analysis whether a species was present and it could have contributed DNA to a sample, by virtue of having been close to that sampling location, or whether or not the DNA from that species is not detected. So it's presence absence. What we cannot do is tell how many of that individual was in that particular habitat. We can actually quantify the number of DNA copies in a sample. That's not terribly hard, but the challenge with quantification is those copies of DNA in an environmental sample could have been contributed in the case of, say for example, fisheries biology, one really large fish or a whole bunch of small fish. It could be contributed by, a bunch of fish further away or one smaller fish right close to the actual sampling location, or it could have been contributed by some members of the overall population that are what we sometimes refers to as super shredders. They're just dumping a lot of their DNA into the environment. So all of those things kind of confound the relationship between the amount of DNA and the amount of organisms in the environment. Next slide. The other aspect that one needs to be aware of is that eDNA doesn't last forever in the environment. So if eDNA is shed into a water column that's well oxygenated, you can assume that DNA, depending on sunlight conditions and other things, is going to be there anywhere from a couple of hours to maybe up to 21 days. As I mentioned previously, if that DNA is captured in sediments that are relatively anoxic, DNA can persist for many hundreds of years, and there's lots of paleoecological studies based on that premise. So that's a limitation, but it's also a benefit. So if I recognize that eDNA has limited persistence in the environment, again, from an aquatic ecology perspective, that's useful to me because I know that if I've actually detected an organism in a sample, that DNA has been contributed relatively recently as opposed to many months in the past. I can start to actually look at seasonal presence. For example, I can look at presence in a small stream in a temperate or subarctic environment under ice versus basically in the summertime open water period. Next slide, please. So what I'd like to do is just run through a couple of examples, and I want to be respectful of everybody's time. So one of the examples that I like, and it's a fairly simple example, has to do with the Pacific water shrew, and some of you will be familiar with this example. Pacific water shrew are listed under the Federal Species at Risk Act. They're present in the Fraser Valley area of British Columbia and a few other locations on the west coast of North America. They're really hard to survey using the previously accepted kind of ecological survey techniques. And the way you do that is you actually go out into the forest. They live, the reason they're called a water shrew is they actually live, the preferred habitat is riparian areas on the edges of creeks and rivers and streams and the like within forested areas. So the way that you survey them is basically you dig some pitfall traps. You those pitfall traps typically in the evening because Pacific water shrew tend to be nocturnal, and then you count the little beasties and you release them or move them, relocate them if that's necessary to protect them, and so on and so forth. The couple of problems with the traditional techniques that have really challenged a whole bunch of people. Number one, Pacific water shrew have really high metabolic rates. So the only effective way to trap them is really pitfall traps. But because of their high metabolic rates, they tend to freak out and they actually because of stress and starvation will die within the pitfall traps very easily, even if you actually had food to the pitfall traps. So, we're trying to basically protect an at-risk species, but our biological survey methods are further putting them at risk, and that's been well recognized. The other aspect of it is to get a really good feel for them is particularly if I know they're potentially in an area and I want to do salvage. Before 2014, Hemmera used to go out and for highway improvement projects and things like that, the requirement was that you'd have to do 8 days of continuous monitoring for each candidate area for relocations. And you basically, those 8 days involved observations at regular intervals throughout the 24-hour period, day and night. So having field biologists out there at all times with some of the attendant health and safety concerns. And again, as I said, that the issue of every once in a while you end up killing one of the shrews rather than salvaging it. So if all in, the salvage costs, we estimate the salvage costs for a couple of projects that we were involved in prior to 2014 were in the range of roughly all in $20,000 to $40,000 per hectare of habitat where we needed to consider Pacific water shrews. The way we've been doing the work since 2014, 2015 is simply looking for the for the DNA of the organism, eDNA, within the waters that the Pacific shrew habitat would be adjacent to. And based on those techniques, we've developed regulatory confidence that we're adequately screening for Pacific water shrew, and those costs are literally 80% lower on a per hectare basis than what we were doing prior to 2014. On top of that, there's zero chance of actually creating incidental mortalities of an at-risk species, and it's basically you can gather the data usually in 1 day of sampling of water rather than 8 days of continuous monitoring, and you reduce basically this health and safety concerns for the field staff involved. Next slide, please. Another example is this particular example, and this is published. You can see the publication there. Jared Hobbs did this work along with our UVic academic partners. This example really shows how sensitive eDNA can be compared to traditional techniques. So next slide, please. Coastal tailed frog, live to be about 25 years. They're in very high elevation, high mountain streams. Habitats you wouldn't think that would be good, very supportive of amphibians. The tadpoles are there for about 4 years, and then they emerge, and then the adults return regularly to the streams. They like to basically find more quiescent areas underneath cobbles as a way of surviving in those kind of high energy environments. And the way that you actually would survey them traditionally is to walk up a stream bed and turn over rocks that you think are good candidate habitat. And so you try to do that gently, but you're inescapably destroying habitat and occasionally causing harms to the particular organisms. So this is a bit busy, this slide, but what you need to know in looking at this map for a sub area of the overall habitat range of tailed frog is that the squares on this map comprise locations that were sampled using time-constrained searches over about a decade and a half prior to 2016. And the circles are locations that were sampled with eDNA, and you'll see there's a lot of the sites that were sampled using traditional methods were resampled with eDNA. The yellow symbols represent areas where tailed frogs have not been detected or were not detected as part of those surveys. And the red symbols reflect those areas where you do see detections. And so what happens is that couple of things. The overall effort prior to 2016 probably comprised somewhere in the range of $300,000 to $500,000 worth of investment. A lot of these areas are helicopter accessible or challenging to get to. It just takes time to actually do the time-constrained searches. Jared was able to sample 50 creeks in 5 days in 2015 for $30,000 all in, and in 2016, I think 69 creeks, again at a cost of around $30,000, including the eDNA analysis. And the important point here is that if you actually look at the eDNA results, and if you look at the time-constrained search results, And Jared's also a tremendously good traditional wildlife biologist, so he was able to confirm his eDNA observations in most cases with really spotting the little beasties in the areas that he was surveying. It turns out that the traditional missing was indeed there. So it shows you the differences in sensitivity. Next slide, please. I'm going to wrap up fairly quickly. Really, this talk was about big picture type of approaches for eDNA. So I want to actually talk about some work elsewhere, and I want to talk about the this particular project is kind of catalyzed by the U.S. Department of Agriculture, the Forest Service, the National Genomic Center for Wildlife and Fish Protection, and for me, it's actually one of the best examples in the world of the potential for eDNA. Next slide, please. This just shows you a map of all of the sites that have been sampled using environmental DNA techniques, the blue and the green dots, throughout the northwestern United States, from Montana through Wyoming, through parts of Washington and Oregon, and down into California, and including up into the Columbia River watershed in British Columbia, although it's not shown here. And so these are samples collected for the purpose of DNA analysis, and U.S. Forest Service, USDA researchers didn't collect all of these samples. What they do instead is they actually provide training and they provide sampling kits, and they create a much far better spatial understanding of the distribution of individual species, in this case bull trout, by actually employing citizen science and creating a common database, so more of a big data type of approach. And there's a bunch of things they're doing with this data, not the least of which is to evaluate the influence of climate change and stream temperature on bull trout and other invasive and native trout species in the western United States. Next slide, please. Let me give one more quick example, and then we'll kind of jump to the conclusions, and my apologies for going a bit long. eDNA has been used extensively in a mining context as well. So I could give you example after example after example. We can spend the next several hours talking about the numbers of different kinds of eDNA applications that are starting to emerge. In the mining space, eDNA has mostly been used for metagenomic studies based on microbial ecology. But some of the examples are to evaluate reclamation success and different microbial groups that change through basically a reclamation sequence for a mine waste or a reclaimed area. Some of the studies have evaluated the role of microbes in acidic rock drainage evolution and a number of other applications like that. Next slide, please, Lori. We'll skip through the next case study here. We'll skip through that one. So important takeaways. Biodiversity protection and enhancement is an increasingly important part of decision-making with regard to new projects and human activities in general. And I think it's fair to say that there's a need now more than ever to really be developing scientific and biological techniques that allow us to look at the scale of the issues over time and over space. And above all, eDNA, as it will continue to evolve, is an important enabling mechanism for really big data approaches for how we're doing ecosystem protection in our ecology. One last slide. Slide, please, Lori. So there is some, I think it's worthwhile to be aware that there is some skepticism about using eDNA approaches as opposed to traditional ecological survey approaches. Part of that is because it's a little bit, because it's developing so fast, it's a little bit feels like the Wild West at times. And so there is a need, there's any number of groups calling for needs for standardization of methodologies and reporting requirements and so on and so forth. So one of the things that Hemmera, along with our current academic partner, Bureau Veritas and our lab Bureau Veritas are doing is to really push for the development, and we're contributing to the development of what will effectively, if it's successful, be the first Canadian and first international standard for DNA through the Canadian Standards Association group. We're also continuing to look for good examples and how to do DNA properly and the associated benefits rather than to actually do it in ways that create greater uncertainty about the value of the approach. So we're working with our UVic colleagues at present to apply for the next round of the Genome Canada Large-Scale Applied Research Program. And what we're looking for is to create a research partnership to look for good case studies where we can really show the true potential of DNA. So if there's anybody today that's actually interested in being involved as an industrial or other type of partner, give me a call after the fact and let me know. Thanks very much. My apologies for running long.
Awesome. Thank you very much, Doug. And I rarely hear anybody complain about a talk like this from Dr. Doug Bright. Hear anybody complain about it going long? So, from my perspective, you're right on time, and appreciate your all of your comments. I will open it up to the question and answer at this point, and I encourage people to use the Q&A box on the right-hand side of your screen. And if you don't see it on the right-hand side of your screen, scroll down to the bottom of your screen where you'll see a menu, and you can click on the Q&A circle, and that'll pop up the box on the right side of your screen. And please enter your question there, and I will ask Dr. Bright. So to get kicked off, Doug, you spoke about, you gave examples of using eDNA for fish. You mentioned amphibians, and you mentioned a mammal. And I guess my question here is, can this be used for any species, or are there limitations on which types of species this is best suited for?
Yeah, I think that the technique is better suited for some species than others. There are actually, the technique has been used for garter snakes. Certain researchers are using it to look for evidence of rattlesnakes. And the way that they're doing that is they're creating boards and habitat that the snake has to crawl underneath, and then sampling that after the fact for evidence of their DNA. So there's any number of things that we've talked about, actually sampling bats. And for example, when you have to assure that endangered and other bats are no longer present when you close a mine adit or a mine portal, for example. And we've talked about sampling the air at a portal entrance, for example, and filtering the air for dust particles and dander particles from bats as one way of sampling. To my knowledge, nobody's done that yet, but I don't see any reason why it couldn't be done. We've talked about sampling black swift, which is an endangered species of swallow, occurs in Banff National Park in a few locations, based on sampling in water, for the simple reason that it's a species that has this unique habit of nesting behind waterfalls and water features. So theoretically, you can test for nest and habitat occupancy by sampling water downstream from candidate nesting sites. The ability, you know, the tendency of the characteristics of the organism or organisms we're interested in relative to the environmental sampling media, you know, gives us clues as to whether or not it's a long shot as to whether we'll be able to detect them with any degree of confidence, you know, or whether it's easier, right? So terrestrial habitats are harder, right? In an aquatic environment, eDNA tends to be transported over reasonable distances, where if there's an organism that I'm interested in within a reasonable distance, chances are I'm going to detect it in the sample, you know, allowing for basically DNA degradation rates and water residence times. In a terrestrial environment, it's a little bit harder because soil doesn't move around in the same kinds of ways. So that's why you'll see most of the applications are related to aquatic species rather than terrestrial species.
Okay, I got it. Interesting. And you mentioned big data and alluded to big data, and that caused a few questions in my mind. And I guess there's probably a series of them here, but is there a consideration potentially of continuous sampling of a medium? And I'm, you know, whether that is air in a cave or an adit setting, whether that's water, either in a allotic or a lentic environment, but regular sampling, and then preserving those samples for and create either testing them or just preserving them for future situations. But is there is there a case has the cost of this gotten to a point where we could have that continuous sampling to create a database over a long period of time, both to monitor for something or even just preserve for situations that we're not even aware of yet? But have we gotten to that point where the cost is low enough and then that sort of study design can be created?
Yeah, I mean, that's a good question, Scott. The nice part about doing eDNA sampling, and I'm going to again talk more about aquatic ecosystems rather than terrestrial ecosystems, right? But yeah, you could do the same thing with filtered air samples as well, right? But in theory, I can actually now sample semi-continuously using something like an ISCO sampler for an aquatic sample for an aquatic environment. Just the way if I'm looking as if I'm looking for total suspended solids, you know, within a watershed, right? So the sample acquisition part's easy. So that's we're certainly actually moving in that direction where you could sample continuously, you know, and almost remotely. Couple of years ago, the genomics technologies were coming down in price, things like high throughput sequencing and qPCR, to the point where people had predicted that the kinds of analyses we're talking about would literally cost pennies per samples. And unfortunately, what's happened in the last probably 3 or 4 years is that the chemicals required to run qPCR, which by the way is exactly the same kinds of technologies we're using to detect COVID. The reagents and chemicals associated with what you're doing in the laboratory became more expensive rather than less expensive. So there was a hope at one point, going back 5 years ago to 10 years ago, that the actual cost of the molecular biology part of this would be so low that you could put it in the hands of everybody and do massive monitoring. Unfortunately, where we're at right now is as the costs have crept up a little bit. So even though the costs tend to be on a comparable basis cheaper than doing ecological survey methods, they're not cheap enough that you can really do massive continuous monitoring yet.
Okay. So that answers a few of the questions that I have here. The next one I have is about the acceptance of eDNA, and I'm wondering, do ultimately regulators and regulators that regulate our clients and our clients' projects, is there universal acceptance of eDNA as a tool across jurisdictions?
Yeah, and good question. So I think I tried to convey the enthusiasm of the academic research community for all things eDNA, whether they're basically community level analyses or targeted taxa analysis. It's really kind of widely recognized that uptake and confidence in the methods by environmental decision makers, regulators, environmental regulators in particular, has significantly lagged behind, right? And so there is certainly a lot of skepticism about the values of these techniques, and more so in some jurisdictions than others. So, for example, because there have been lots of good examples in the United States, those U.S. Department of Agriculture, Forest Services folks that I was making reference to before, don't understand why we Canadians are actually so slow in terms of the uptake in terms of practical applications, right? And similarly, the US Geological Survey, and there's some other good examples there. There's some really good examples over in Europe and the UK. In British Columbia, there's greater uptake because really in Western, there's a couple of birthplaces of eDNA practical applications in Canada. One is in Ontario out of the University of Guelph, and another one is, in British Columbia, based on the earlier work that Jared Hobbs and was doing, working directly with BC government ministry folk on those kinds of at-risk species studies. You go to Alberta, there are very few good examples that people can point to and say, yeah, I think this really works. If you go to Saskatoon, there are even fewer. So regulatory acceptance requires transparency and simple decision making, and that in turn requires that all of the people doing eDNA work compromise and adapt some kind of standardized methodologies, so that everywhere you see an eDNA study, you're kind of comparing apples with apples rather than apples with oranges. And that's something that is there's a large call for it as an important prerequisite to getting larger kind of regulatory and environmental decision maker acceptance of eDNA, but we're just starting down that path. So I mentioned the CSA initiative as one of the kind of many things that are needed to get us there.
Right. I've got one question from the audience as well. Doug, you indicate being able to detect presence, both present and past, but can you also determine the scale in varied time periods? So, i.e., how many shrews or species at a given point in time, either present or past?
Well, and you know, I think most of the studies around historical reconstructions are based on just minor extensions of what is referred to as paleoecology, right? So they're largely based on lake sediment core studies. So what I've got is I take my core, I section it, I do basically lead-210 and cesium dating so that I actually know when the sediment was laid down historically. Geological observations. You know, historically, researchers might have looked for things like diatom frustules or the remnants of plants in terms of plant spores and things like that. And now those studies are simply looking for the DNA of the organisms rather than remnant biological structures. So in that context, it's really easy, right? There are probably some other aspects where you can do historical reconstructions, but I'm not aware of many other than doing basically the core studies in lake and marine ecosystems.
Makes sense. Okay, thank you. Well, I think we're at 9:46 now in Vancouver, and this has been a great informative talk, as it always is listening to you speak. I would remind the audience that your on the slide on the screen, your email is there. And if the audience has further questions or would like to know more about this, or potentially is interested in deploying eDNA on one of their projects, to please reach out to Doug. And with that, I'm gonna wrap up this week's Ausenco Connect webinar. Thank you very much to everybody for joining. Thank you very much to Dr. Doug Bright for the time to talk about this, and we look forward for you to joining us on our next Ausenco Connect. So thank you very much, everybody, and have a great day.